Our lecture today is about embryology and embryogenesis. The embryogenesis is a process of formation and development of the embryo, including human embryo, and the embryology, the medical embryology. It's a science which is about embryogenesis with research processes and rules of development of the embryo. Knowledge of medical embryology necessary for formation of medical meaning in future doctors. Individual development of the embryo begins since moment of formation of the unicellular organism till death, and this period is named ontogenesis. Ontogenesis is divided into two main periods: prenatal and postnatal. Prenatal, which is since fertilization till birth, and postnatal, which is since birth till death. Prenatal period of the ontogenesis is divided to next periods. The first one, it is for since first till second weeks is named the beginning period, and next one since third till eighth weeks of development, embryonal period, and the last one it's a fetal period since 9th till 38 weeks. For first and the second period, organism is named embryo. For next period, last one, it's named fetus. And development of the human embryo begins since the moment which is named fertilization, when two sex cells fuse together with formation of the diploid unicellular organism which is named zygote. Two sex cells take part in the human fertilization: male sperm cells and female oocyte. Each sex cells contain haploid chromosomal set, which include 23 chromosomes, 22 of which are autosomes, and one sex chromosomes. Sperm cells contain X, R, Y, Y sex chromosomes, but oocyte contain only X chromosomes. So, sex of child is determined by the sperm. Process of formation of sex cells is named gametogenesis or progenesis. In mean, it lasts for all of all lifetime since puberty period. In women, gametogenesis begins before birth and lasts till 50 years. Human sex cells, sperm, are formed in convoluted seminiferous tubules of the testes. The mature sperm has about 65 micrometers, long about 65 micrometers. It's composed by head, neck, and tail. The tail has three parts: middle piece, principal piece, and the end piece. Membrane of the anterior part of the head has the receptors to the ovum, which are covered or closed by the protective protein cap. Also, plasma lemma of the sperm contain a special enzyme, glycosyl transferase, which is necessary for recognizing of the ovum. And this cap on the apical or anterior part of the head, sperm cells has a chromosome. It's modified Golgi complex, Golgi apparatus, which systems filling enzymes which necessary for penetration layers or tunics of the ovum. Within enzymes present proteolytic enzyme acrosine, which is a neutral proteinase, and the hyaluronidase, trypsin, and other. Nucleus of the sperm take 2/3 of the volume of the head. The neck of sperm contains two centrioles and their spirally located mitochondria. Proximal centriole is the closest to the nucleus and take an active part in fertilization. The distal centriole is hollowed with microtubules of the axoneme complex. Axoneme of the tail is composed of the microtubules, which have a pattern of location 9 multiplied 2 plus 2. By the structure, it's a flagella, it's a special organella. The middle piece of the tail is covered by the mitochondrial sheath, which give energy for movement of the sperm. The speed of movement of sperm inside in a female reproductive vase is about 50 micrometers per 1 second. The direction of movement of sperms is directed by distribution of special substances which are produced and released by follicular cells of the oocyte. And the ability of sperm cells to move toward the source of distribution of these special substances is named chemotaxis. The female sex cell ovum is formed in the ovary as the oocyte. It continues to be oocyte till fertilization. After liberation from the ovary in ovulation, it continues to be oocyte, secondary oocyte, and can become the ovum only at the moment of fertilization, and particularly at the moment of penetration by the sperm layers of the oocyte. Is here a classification of ovums. Depend on the amount. It depends on the amount of yolk inclusions and depend on the distribution of yolk inclusion in the cytoplasm of ovums. Main function of yolk inclusion it's the give nutrition, trophic function for future embryo. Depend on the amount of yolk inclusion, all of ovums can be classified to elysital, we don't contain any yolk inclusion. Oligolecytal, which contain a small amount of yolk inclusion, a little bit, and polylecytal, which contain a large amount of yolk inclusion, like birds. Oligolecytal, like human ovums, or lancelot, and in the phylogenetic set of human ovum go by the lancelot ovum, so that we call them secondary oligolecytal. Depending on the distribution of yolk inclusion, ovums can be classified to isolecytal, where yolk inclusion are distributed everywhere in the cytoplasm of the ovum, and telolecytal, in the cases of formation of aggregation of yolk inclusion near one of pole of the ovum, like in birds. So, depend on the classification, human ovum is secondary oligolecytal and isolecytal. Secondary oocyte has a round shape and it has diameter about 130 micrometers. It's surrounded by the zona pellucida and follicular by the layer of follicular cells which form corona radiata. Zona pellucida, it's an acellular structure which mainly composed by glycosaminoglycans and hyaluronic acid, which are produced by follicular cells. And corona radiata is composed by processes of follicular cells which connect with the cell membrane of the oocyte and provide nutrition to the oocyte. Oocyte actually need for this connection, and they need this nutrition because oocyte don't connect with blood vessels, but it for maturation, it's for living, it need nutrition. And oocyte, which is located inside in layers of follicular cells and zona pellucida, can get nutrition only from follicular cells. Cytoplasm of the oocyte filling rough endoplasmic reticulum, molecules of RNA, there are a lot of ribosomes, but doesn't contain centrioles. And peripheral part of the cytoplasm contain cortical granules, which chemical structure is a proteoglycans and glycoproteins, which after penetration of layers of the oocyte and after penetration of the cell membrane of the oocyte by the sperm, create impenetrable layer in the perivitelline space, which is located between zona pellucida and cell membrane of the oocyte. And this impenetrable layer prevent polyspermia. Fertilization is a process of fusion of the ovum and sperm, which result is formation of the unicellular organism, which is zygote. Process of fertilization pass three main stages. The first stage is a distant stage of relation between gametes. It happens due to chemotaxis. The second stage is a contact stage, which is releasing of acrosome reaction with next penetration of layers of the ovum. In this advanced third stage, it's a penetration of sperm and activation of the ovum. Will happen in region of the head and the neck of sperm into the cytoplasm of the ovum, and with activation of the cortical reaction. Process of fertilization happen in the upper part of the uteral tube, which is the longest and widest part of the uteral tube. And the 6 and 7 hours here happen capacitation. It's happen with sperm. It's a chemical modification of cell membrane and acrosomal membrane of sperm. As a result of which changes in the cell membrane of the sperm, which help to future cortical reaction, to future liberation of enzymes of the acrosome. Releasing enzymes of the acrosome is named acrosomal reaction. It's necessary for penetration of layers of the oocyte. I mean, corona radiata and zona pellucida. For penetration, for penetration of zona pellucida, I used proteolytic enzyme, and for penetration of the corona radiata, excuse me, for penetration of the zona pellucida is used hyaluronidase. And the next step is the penetration of the cell membrane of the secondary oocyte. As you see in this picture, the head and the neck of the sperm enter to the cytoplasm of the oocyte, and the acrosomal reaction at this point finished. As answer on the penetration of the cell membrane, the cortical reaction begins. Cortical reaction, during cortical reaction happen liberation of cortical granules from the peripheral part of the cytoplasm of the secondary oocyte to the perivitelline space, which is located between the cell membrane of the secondary oocyte and zona pellucida. And here happening denaturation of proteins of the zona pellucida under the influence of enzymes of cortical granules, and with formation of unpenetrable layer in the perivitelline space. It's for preventing of the polyspermia. After that, happen a fusion of two pronucleus of two gametes, I mean ovum and the sperm. Before fusion, before finish penetration, the secondary oocyte finished second meiotic division and became the ovum. After coming the sperm pronucleus into the cytoplasm, happened fusion to pronucleus of sex cells, and the cardiolem disappear, and chromosome material fuse together with formation of Synkarion. And this process of fusion of Synkaryons of two sex cells in fertilization is named syngamia, and syngamia finish the fertilization. And next, some days after fertilization, happen set of mitotic divisions, which is named cleavage. We call this mitotic division as cleavage because of very short interphase. And next generation of cells became smaller and smaller and smaller. So having increasing number of cells without increasing of size. Size of next generation is small decrease. And the name of each cell, it's a blastomere. Um, the type of human cleavage, it's a complete, subequal, and or unequal and asynchronic. And a next type of organism which forms after zygote, it's a morula. Morula is a multicellular organism which doesn't contain inside space. After morula, we get blastocyst. Blastocyst is a multicellular organism which contain inside space. Morula made up of 8 till 12 blastomeres, and the blastula blastula is composed by 16 blastomeres and contains space inside with filling fluid, which is really produced and released by cells by blastomeres. As you see here, as a result of an unequal division or unequal cleavage, human blastula has outer small light cells and inner large dark cells. Inner large dark cells give rise to the embryo body, and this mass of cells is named embryoblast. And outer small light cells are named trophoblast. This layer of cells will give rise to the placenta. The process of cleavage is followed by movement of the blastocyst toward the embryo, following by the movement of embryo toward the space of the uterus. And on the stage of the early blastocyst, the embryo is located in the space, enter to the space of the uterus on the fourth day after fertilization. On the fifth day, happen Getchung. Catching its loosing of the fertilization shell. Because of functional activity of cells of the trophoblast, they start to release enzymes which break the fertilization shell. It's very important. It's very important for future implantation.